Hello everyone, welcome to Evolutionary Biology class. We are in Unit 2, which is Molecular Evolution, and today's topic is Protein Synthesizing Machinery. So, in introduction, ribosome is the site of protein synthesis. So, tRNA brings the desired amino acids, reads the genetic information, and places the amino acid at the proper place. So, RNAs are formed from DNA during transcription, during the process called transcription, while protein synthesis occurs in the cytoplasm over ribosomes. So, these two are separated both in space and time because transcription occurs in the nucleus and the protein synthesis occurs in the cytoplasm. So transcription and translation occurs in separate space and time, so because it prevents the intermixing of different raw materials, and it also protects the DNA from respiratory enzymes and ribosomal machinery from different nucleases. And next is machinery for protein synthesis. So for protein synthesis, different types of machinery that is required consist of ribosomes, amino acids, mRNA, tRNAs, and amino acid tRNA synthetases, that is an enzyme. And second is mRNA functions as a template having the genetic information. So let's see these different machinery parts one by one. So first one is ribosome. Protein synthesis occurs over ribosomes. So ribosomes are therefore called the protein factories. And each ribosome has two unequal parts. One is large and one is small. The large subunit of ribosome has a groove for pushing out the newly formed polypeptide and protecting the same from cellular enzymes. So here we can see the picture of the ribosome. So one is the bigger subunit and the smaller subunit. So these ribosomes basically acts as a protein factory. So in figure 1 we can see the relationship between the various components, and in figure B we can also see the synthesis of polypeptide on ribosomes, which is connected with the endoplasmic reticulum. So further, the smaller subunits fits over the larger one like a cap but leaves a tunnel for mRNA. This means that the smaller subunits acts as a cap over the larger subunit. The two subunits come together only at the time of protein formation, and this phenomenon is known as association. So for the process of association, that is for the two subunits to come together, magnesium ion is very essential for it. And soon after the completion of protein synthesis, the subunits again separate from each other, and this separation is called as dissociation. Ribosomes usually form rosette or helical groups during active protein synthesis, as seen in the picture below, and these multiple ribosomes are also known as polyribosomes or polysomes, which was defined by Rich in 1963. The different ribosomes of polysome are held together by a strand of messenger RNA or mRNA. So polyribosomes helps to produce a number of copies of the same polypeptide at once, and that the adjacent ribosomes or a polyribosome are about 340 angstrom or 34 nanometer apart. So when the ribosomes, multiple ribosomes in a polyribosome come together, the distance between two ribosomes is around 340 angstroms or 34 nanometer. Okay. The different parts of ribosome connected with protein synthesis are: number one is a tunnel. There is a tunnel between for the mRNA to come and lie. So it lies between the two subunits, and there is also a groove for passage of newly synthesized polypeptide. The groove is part of a larger subunit, and there are altogether three active sites, which is P or D, A and E. So P, A, E. So there are three. These are the three active sites. P site is also known as peptidyl transfer or donor site. That's why either it is P site, so that is peptidyl transfer site, or the donor site. That's why it is known as either P site or D site, which is jointly contributed by the two ribosomal subunits. A site is also known as amino acid or acceptor site, which is situated on the larger subunit of ribosome, and it faces the tunnel between the two subunits. And finally, there is a E site or an exit site, which is a part of a larger subunit facing the tunnel. So this is the ribosome and the different active sites. So the A is the acceptor site, P is the peptidyl transfer site, and E is the exit site. So enzyme peptidyl transferase is a ribozyme. So it is a component of a larger subunit of ribosome 23 sRNA in a prokaryotes, and a smaller subunit of ribosome has a point of recognizing mRNA and binding area for the initiation factors. So second part of the protein synthesizing machinery is the amino acids. Hundreds of different types of proteins can be manufactured in a single cell, and all types of proteins are formed from the same amino acids, which are 20 in numbers. So it is therefore the arrangement of the amino acids in the polypeptides and the number of the latter, that is of the same amino acids, which provide the specificity to the different proteins. So there are some 20 amino acids and amides which constitute building blocks or monomers of proteins, and they occur in the cytoplasmic cellular pool. So the third part of the protein synthesizing machinery is the mRNA or the messenger RNA. So mRNA is the messenger RNA which brings coded information from DNA and takes part in its translation by bringing amino acids in a particular sequence during the synthesis of the polypeptide chain. However, the codons of the mRNA are not recognized directly by the amino acids, but it is being recognized by the anticodons of their adapter molecule, and the translation of these mRNA messenger transcript occurs over the ribosomes, and the same messenger RNA which brings the coded information from DNA is can be reused multiple times. So it can be used time and again, and in the form of polysome it can help synthesize a number of copies of the polypeptide chain simultaneously. So under mRNA, let's also discuss a topic known as RNA processing. In eukaryotic cells, the newly made mRNA molecules undergo a series of modification which is known as RNA processing. So enzymes in the cell add a special nucleotide called a cap at one end and the string of adenine nucleotides called a tail at the other. But the cap and tail help protect the mRNA from being degraded in the cell. So the mRNA molecules also undergoes a process known as splicing, which is an elaborate cutting and pasting procedure. So the segments of the newly made mRNA called introns. So the introns are cut out and discarded, and the remaining mRNA segments, which consist only of exons, which are the express sequences, are kept and linked together. So once the introns are cut and discarded, the final mRNA is also known as the mRNA transcript. So splicing is the work of a RNA and protein complex known as the spliceosome, and only once the splicing is complete, the mRNA is ready to be translated into a protein. So here we can see how from a DNA pre-mRNA is being formed, which consists of both the exons and the introns part, and the spliceosome helps to discard and cut or splice the introns away, and finally the mRNA transcript, the final mRNA transcript which is required for protein coding is then formed, and only the final mRNA transcript is then used for further translation of the proteins. The next important part of the protein synthesizing machinery is the tRNAs, also known as the transfer or soluble RNAs, which pick up the particular amino acids as the CCA or the 3' end in the process called charging. The charged tRNA take the same to mRNA over particular codons corresponding to their anticodons. A tRNA can pick up only a specific amino acid, although an amino acid can be specified by 2 to 6 tRNAs. So each tRNA has an area of coming in contact with ribosome and the enzyme amino acid tRNA synthetase. And the fifth part of the protein synthesizing machinery is the amino acid tRNA synthetase. So what is amino acid tRNA synthetase? It is a enzyme that helps in combining the amino acid to a particular tRNA, and the enzyme is specific for each amino acid, and it is also called AA activating or the amino acid activating enzyme. So we have dealt with the different parts of the protein synthesizing machinery, and now let us see the mechanism of the protein synthesis. First one, the first step in the mechanism of the protein synthesis is the activation of the amino acids. So the activation of the amino acid is carried out by activating enzymes known as the amino acid tRNA synthetases. So it was first been discovered by Jemnik and Hoagland in 1957. In the presence of ATP molecule, an amino acid combines with its specific amino acid tRNA synthetase, and here for this process magnesium ions is being required. So it produces amino acid adenylate enzyme complex, and this is also been shown in the equation here, where AA is the amino acid in the presence of ATP plus enzyme, which is amino acid tRNA synthetase in the presence of magnesium ions, produces the complex. The energy made available to amino acids during its activation is also later used in the formation of peptide bonds. So the hydrolysis of pyrophosphate with the help of enzyme pyrophosphatase provides energy for driving the initial reactions. In the second part of the first step only is the charging of amino acylation of tRNA. So the complex reacts with tRNA specific for the amino acid to form amino acid tRNA complex, that is which is specific for specific amino acid. So enzyme and AMP are released. The tRNA complex with amino acid is sometimes also called the charged tRNA. The amino acid is linked to the 3' OH end of the tRNA through its carboxyl group. So this is the figure which shows the possible steps involved in synthesis of polypeptide from amino acids through the agencies of different tRNA, mRNA, amino acid synthetase and ribosome. So the second step in the mechanism of protein synthesis is the initiation. It requires factors called initiation factors. So there are three initiation factors in prokaryotes, namely IF3, IF2 and IF1, whereas in the eukaryotes they have 9 initiation factors, which is E, IF2, E, IF3, E, IF1, E, IF4A, 4B, 4C, 4D, E, IF5, E, IF6. So these are the 9 initiation factors in the eukaryotes. So out of these, initiation factor 3 or the EIF2 in the eukaryotes is attached to the smaller subunit of ribosome in the dissociated state. So for the process of initiation, GTP is required, and the mRNA attaches itself to a smaller subunit of ribosome in the region of its cap. The cap has nucleotides complementary to the nucleotides present at the 3' end of the ribosomal RNA, that is the rRNA. The attachment is such that the initiation codon of mRNA, that is the AUG or the GUG, comes to lie at the P site, and the initiation factors already present in the smaller subunit catalyzes or the fast forwards the reaction. Next, the amino acid tRNA complex specific for the initiation codon, that is the methionine tRNA or valent tRNA, reaches the P site, that is the peptidyl transfer site or the donor site, and the anticodon, that for an example UAC of tRNA methionine, establish a temporary hydrogen bond with the initiation codon, that is the AUG of the mRNA. The codon-anticodon reaction occurs in the presence of a initiation factor EIF3 in eukaryotes and IF2 in prokaryotes. This step also requires energy, which is provided by the GTP. So it is also been shown by the equation here. And the initiating methionine accepting tRNA is charged with non-formulated methionine in the cytoplasm of eukaryotes and formulated methionine in prokaryotes, plastids and mitochondria. The tRNA engaged in transferring formulated methionine is different than the one that transfers non-formulated methionine. So in the presence of magnesium ions, the larger subunit of ribosome now combines with the 40S mRNA tRNA methionine complex to form intact ribosome. So it requires initiation factor 1, that is IF1 in prokaryotes, or the factors EIF1, EIF4, A, B and C in the eukaryote. So the coming together of the two subunits of this ribosome is also known as association. The intact ribosome now encloses the mRNA tRNA complex present at the P site, but it keeps the A site exposed. So the next step in the protein synthesizing machinery is the elongation. Elongation is also known as the formation of polypeptide or the polypeptide chain formation. An amino acid tRNA complex reaches the A site and attaches the mRNA codon next to the initiation codon with the help of its anticodon. The step requires GTP and that is an energy and an elongation factors EIF1 in eukaryotes and EAF-TU as well as EAF-TS in prokaryotes. It has been found that in Escherichia coli, the most abundant protein is the elongation factor EAF-TU. A peptide bond is established between the carboxyl of amino acids attached to tRNA at P side and amino group of amino acid attached to the tRNA at the A side. In the figure given below, the process of elongation of polypeptide chain is been also shown here. The reaction is catalyzed by enzyme peptidyl transferase, which is an RNA enzyme. Due to this, the amide group of the first amino acid is blocked from getting involved in the peptide bond formation with another amino acid. So in the process, the connection between tRNA and the amino acid at the P site breaks. The free tRNA of the P site slips to E site and from there to the outside of ribosome with the help of the G factor, and the A site carries the peptidyl tRNA complex. Soon after the establishment of first peptide linkage and slipping of the free tRNA of P site, the ribosome of mRNA rotates slightly, and the rotation of the mRNA is also known as translocation. It requires a factor called translocase, which is EFG in prokaryotes and EEF2 in eukaryotes, and it also requires an energy in the form of GTP molecule. As the result of translocation, the A site codon along with the peptidyl tRNA complex reaches the P site. A new codon is exposed at the A site, so it attracts a new amino acid tRNA complex. The process of bond formation and translocation is repeated, and one by one all the codons of mRNA are then exposed at the A site and then get decoded through incorporation of amino acids in the peptide chain. So the peptide chain elongates. The elongated peptide chain or the peptide polypeptide, also known as the polypeptide, lies in the groove of the largest subunit of ribosome to protect itself from various cellular enzymes because it is prone to breakdown due to its extended nature. The helix formation begins at a distance with the help of chaperones, and a lot of energy is consumed in the formation of protein or in the process of protein synthesis. Therefore, for every single amino acid incorporated in peptide chain, 1 ATP and 2 GTP molecules are used, and finally the process of termination takes place. So the polypeptide synthesis is terminated when the nonsense codon of mRNA reaches the A site. So there are three nonsense codons as we have discussed in the previous class, which is UAA ochre, UAG amber and UGA opal. So these codons are not recognized by any of the tRNAs. Therefore, no more amino acid tRNA reaches the A site. The P site tRNA is hydrolyzed and the completed polypeptide is released in the presence of GTP dependent release factor. So the release factor is a single ERF1 in eukaryotes, and two release factors is required, that is RF1 and RF2 in the prokaryotes. In prokaryotes, RF1 is specific for UAG and UAA stop codons, whereas RF2 is specific for UAA and UGA. GTP dependent RF3 or ERF3 in yeast is required for releasing the releasing factors from the ribosome. So ribosome moves over the nonsense codon and slips of the mRNA chain. The two subunits of ribosome and then finally separate or undergo the process of dissociation, and in the presence of dissociation factor known as DF. In prokaryotes, formulated methionine is commonly the initiating amino acids. It is either deformulated with the help of enzyme deformulase or sometimes removed from polypeptide by the enzyme aminopeptidase. The initiating methionine is usually not retained in the eukaryotes. So at a time, several polypeptides are synthesized from the same mRNA by a polyribosome, where a number of ribosomes are attached to the same mRNA strand, as we have seen in the pictures before. So each ribosome of a polyribosome forms the same type of polypeptide. Formation of a number of copies of the same polypeptide simultaneously from an mRNA with the help of polysomic is called translational amplification. On being released from the ribosome, a polypeptide has only a primary structure, and after that it coils and folds further to have a secondary and tertiary structure. A polypeptide may get associated with other polypeptides to produce, that is, a I-pleated structure, which then forms tertiary and quaternary structures. Or in the case of rich cytoplasmic polyribosomes, the polypeptides or proteins are released in the cytoplasm or the cytosol, where they are now employed for synthesis of more cytoplasm. Some enzymes and components of cell organelles like nucleus, microtubules, microfibules and microbodies according to the proteins formed. So some proteins also enter the composition of semi-autonomous organelles like plastids and mitochondria, though they manufacture a part of their protein requirement themselves by their own polyribosomes. So the polyribosomes attached to the membranes of endoplasmic reticulum produces proteins which either pass into its lumen or become integrated into its membranes. So the proteins released into the endoplasmic reticulum lumen generally reaches the Golgi apparatus for various modifications like formation of hydrolytic enzymes and glycosylation, which is the addition of sugar residues. And these modified proteins are then finally packed in vesicles for export or for the formation of lysozymes, cell wall enzymes or plasma membrane and other things. So the protein synthesis is inhibited in bacteria by certain antibiotics, and this forms the basis for treatment of bacterial infections. And in this slide we can see the different classes of antibiotics and their mode of functions or inhibitions of bacterial protein synthesis. So in short, if let us see the animated video of the protein synthesizing mechanism. So ribosome is a large molecular factory that translates the genetic code in the mRNA into the long chains of amino acid that folds up into a protein. The genetic code is read in a group of 3 letters called a codon, as we have already discussed in the previous class. Amino acids are transferred to the ribosome by tRNA or the transfer RNA, which match the gene code in the mRNA. So once the gene translation is complete, the amino acid chain folds up to form a protein in a 3-dimensional shape that determines its various functions. And by this we come to the end of the topic. Thank you for listening. Please like, share and subscribe to the channel for the upcoming classes. Thank you.